In 1665, Robert Hooke discovered the cell. After years of research, scientists have put together the cell theory. The cell theory states that all living things are composed of cells. Cells are the basic units of structure and function in living things, and new cells are produced from existing cells. Cells have been divided into two different categories: prokaryotic cells and eukaryotic cells. Prokaryotic cells have cell membranes and cytoplasm, but do not have nuclei. Eukaryotic cells have cell membranes and cytoplasm and do contain nuclei. The nucleus is like the brain of the cell. It controls most of the cell's processes and contains DNA. The nucleolus is a small dense region inside of the nucleus. The nucleolus is also where the assembly of ribosomes begins. Ribosomes aid in the production of proteins within the cell. The nucleus is surrounded by a double membrane layer called the nuclear envelope. The nuclear envelope is dotted with thousands of nuclear pores, which allow materials to move into and out of the nucleus. The nucleus sends a steady stream of RNA and other information-carrying molecules to the rest of the cell through the nuclear pores. The granular material visible within the nucleus is called chromatin. This consists of DNA bound to protein. When a cell divides, chromatin condenses to form chromosomes. The endoplasmic reticulum is the organelle in which components of the cell membrane are assembled and some proteins are modified. The part of the endoplasmic reticulum known as rough ER or rough endoplasmic reticulum has the name because of the ribosomes that stud its surface. The smooth ER, which does not have the ribosomes on the surface, contains collections of enzymes that perform specialized tasks such as the synthesis of lipids. Proteins produced by the rough endoplasmic reticulum move to a stack of membranes called the Golgi apparatus. The Golgi apparatus was named after an Italian biologist, Camillo Golgi, who discovered it. Enzymes in the Golgi apparatus attach to carbohydrates and lipids to proteins. From the Golgi apparatus, proteins are then sent to their final destinations. Many plant cells have large sac-like structures known as the central vacuole. The central vacuole holds liquids like water, salts, and proteins. Chloroplasts are found in plants and some other organisms. Chloroplasts use energy from sunlight to make energy-rich food molecules in the process known as photosynthesis. Mitochondria is found in nearly all eukaryotic cells, including those of plants and algae. Mitochondria use energy from food to make high-energy compounds that the cell can use to make power, growth, development, and movement. The cytoplasm is the material inside the cell membrane, but not including the nucleus. Microtubules are hollow tubes of protein about 25 nanometers in diameter. They maintain cell shape and can also serve as tracks along which organelles are moved. Microfilaments are long, thin fibers that function in the movement and support of the cell. Cilia and flagella can produce considerable force. In some cells, cilia move against like the oars of a boat, pulling or pushing cells through the water. Vesicles are smaller vacuoles used to transport things within the cell. The main job of the cell wall is to protect and support the cell and everything inside. The cell membrane is a selectively permeable membrane that regulates what enters and leaves the cell, whether it's from diffusion, which is the movement of a solute from a high concentration to a low concentration across the cell membrane until equilibrium is reached, or osmosis, which is the diffusion of water. Facilitated diffusion is next. It's where glucose is helped into the protein channel. Lastly, active transport, which is basically facilitated diffusion, but it needs energy in the form of ATP.